You have that Mediterranean diet with, a good amount of vegetables, good amount of protein.
Yeah.
All has to be well sourced, of course, and some good fats. And then some people are going to do better with more vegetables. Some are going to do better with more protein.
That's right.
And maybe there's different points in your life where you do better on one and the other one too, right?
That's a very good. Yeah, I mean, if you're doing a bunch of high intensity and interval training, you're going to need some readily accessible carbs.
Right.
If you kick those to the curb and you're just doing, something with almost no carbs like carnivore, you're going to feel that when you really try to hit those heights with exercise. So to your point, yes.
Dr. Fitzgerald, I'm so excited to finally have this conversation with you. We've been friends for a while now.
A long time,
And this is, this is so great to get you on the podcast to talk about your program and the Younger You program in general, but also all the research you're doing and how it's evolved over the last few years. It's going to be a huge conversation for me for this podcast, especially because I've been through it myself. I've taken patients through it.
Yes.
And I'm excited to talk about it with you.
Awesome. I'm really happy to be here. Anytime getting to hang out with Dr. Shaw is a good time. It's time well spent.
Thank you. So, Dr. Fitzgerald, should I call it Kara? Or Dr.
Of course,
Kara?
Kara.
Okay. Kara, so, your evolution through this is really interesting. I'd love for you to talk a little bit about your practice, kind of how you started, and how you happened upon doing your study on the reversal of biological age. Would you talk a little bit about that?
Yeah, it's a pretty interesting story, and I can get into it and get really, really obsessed about it because it's interesting to me as just unpacking it over the years. But feel free to interrupt me at any time. I started to think about epigenetics in earnest in around 2013. A lot of papers were crossing my desk, most of it about cancer. So, and we can circle back to this later, but cancer aggressively hijacks epigenetics and gene expression, aggressively. And being a functional medicine clinician in a teaching clinic, the big question nagging me was how could functional medicine, this is systems medicine, this is whole person medicine. How is what we are doing influencing gene expression through epigenetics? And because the papers on cancer were the primary ones crossing my desk at the time, my question was, simply, are we benefiting? Are we favorably influencing this? Could anything that we're doing be negatively influencing cancer's ability to take over gene expression? So that was the first question. And with my very smart nutrition director, the director of our nutrition programs at the clinic at the time, Romilly Hodges, we dove into the science. And there wasn't a lot of science. I mean, that has changed incredibly. A lot of what we were looking at were cell studies, animals if we were lucky. And we saw that diet had a profound influence on gene expression. In animal studies, in cell studies.
Because when you were looking at these papers, looking at the methylation of the DNA was something that was rarely done in humans and not at a mass level for sure.
No, I know.
So how are you even checking these? How are they back then running these methylation studies? Was it a specialized machinery or specialized labs?
Well, Illumina, kind of the flagship lab, had smaller arrays back then. Okay. So there's, 30 million methylation marks on the genome. There's a lot of them. And back then they were like 27,000. I mean, they were they were smaller. They were a little bit more cumbersome. They were a little bit less reliable, but they were there. People were able to do it. Yeah, but most of the data were in cell studies and in animal studies.
So you're looking at all these studies, and then how did that jump into you actually doing a human study?
So it's such a great question. So from our read on the literature, we put together our best hypothesis on what an optimal diet and lifestyle program would look like to change favorably DNA methylation. Wow. So we took all of that literature. There were scant human studies, like some in exercise, there were some in meditation, but really the bulk was in animal. And we put together the diet and lifestyle, the methylation diet and lifestyle program that we then rebranded Younger You, but that's the name that we use in research, methylation diet and lifestyle. And we started to do it in practice, but we couldn't measure epigenetics at the time. There were no tests to send a specimen into. It was only in the research setting, but we could see patients getting better. We could see people like responding. We could see their antibodies numbers dropping if they had autoimmunity. We could see their inflammation numbers dropping. They would lose weight. I mean, we routinely saw people really responding clinically to this dietary pattern over and over and over again. One of our doctors always says it's as impactful for her as the standard elimination diet that we do in functional medicine for, many, many years. So we could see clearly that it was a powerful intervention. But we didn't know if it was changing epigenetics, if it was changing gene expression. And it was a conversation that I had with the then CEO of Metagenics, supplement company, around some of our questions around using high-dose B vitamins and whether that could negatively influence gene expression. So we were coming at it through that angle, and it grabbed his attention. In fact, at that time, his wife was pregnant, and she was taking a very high dose, B vitamin prenatal, as we always prescribe, and as is warranted. But knowing that there were some data out there that potentially challenged how and when we used these doses, just caused him to pause profoundly, enough so that he funded us. Metagenics gave us an unrestricted grant. They said, you can do this, Kara. You'll own what you guys identify. You can design the study. He put us in the driver's seat entirely. So we were able to hire a clinical research center, actually the Health Gut Research Institute at my alma mater, National Institute of Natural Medicine, to run a randomized control clinical study. Wow. And in that setting, we were able to look at epigenetics. We could, order all of our test kits from Illumina directly and so on and so forth. So that set it off. Biological age at that time, Darshan, was thought to be irreversible, like that you couldn't influence it with interventions. That's what Steve Horvath really believed and said. In fact, I saw him lecture at PLMI, saying, I heard him many times just really talk about us not having much power over influencing the direction or, changing our biological age. So even though we were going to have the tools to be able to measure that, I didn't know, I wasn't holding my breath. I was hopeful that we would favorably change gene expression, but I really didn't know what and how, because again, this was the first of its kind study. Right. It was by the grace of God we happened to get funding to go in and do this.
That's amazing.
Yeah. So we went in with, we're hoping we're going to change gene expression towards something better. And during the study, it took us a year to run it. The first paper came out showing biological age reversal in humans, and that was the TRIM study in, where is he? He's up near Stanford, Greg Fay and his group. And they had a year-long intervention using growth hormone, metformin, DHEA, zinc, and they were able to reverse biological age. And he continues to, research his protocol and refine his protocol and, just get better and better. But time stood still. So those of us doing research in epigenetics were really excited about that study. It was extraordinary. So, and Steve Horvath, the guy who developed the clocks, was was an author on that paper. I think he was the first author, and or he was the last. Well, anyway, he shared it with Greg, and he did all the clocks, and so there was just this clear bioage reversal, and it was very, very exciting.
Was he also surprised by the result?
Yeah, 100%.
He was just mind-blown.
100%. Yes.
And this set off a whole new field. It set off this entirely new direction.
Yes, that's right.
Of, I would say, healthcare now. You know, if you really look at this.
And it was just a few years ago.
Right. Yeah. When was the TRIM study?
2019.
2019. And your study was published when?
2021.
Wow.
In between those, or it was actually a little bit before, but it was a little bit before Greg's study, but there was a vitamin D study, and they showed that D deficiency was associated with an accelerated biological age. And when you repleted vitamin D levels, that the acceleration slowed down. It wasn't a randomized clinical trial in the way that, we were doing, but it was able to show deficiency associated with age acceleration and then repletion, slowing it back down.
I love that. And so you saw this data, you got the funding, and back then there were no companies doing biological age testing except for Illumina sending you these special kits.
They just gave us the kits, and then we had to have we worked with Yale to process all of our study specimen, and Horvath has a calculator at UCLA, so we put our data into his system and used all of his tools to actually calculate our clocks. Yeah, it was very different than it is today.
Right. And the TRIM study was interesting because they use growth hormone. They use some real unconventional, I would say, medical treatments.
Yeah.
Whereas you took a dietary and lifestyle approach to the same problem.
Yeah, that's right. That's right. Yeah. And much shorter time frame, 8 weeks.
8 weeks.
Now, it's an all-in diet and lifestyle, as you know.
Right.
The diet is, it's a healthy dietary pattern by any measure. There's a lot of veggies. There's some fruit involved in it. There's protein, good fats, a gentle intermittent fasting structure, 12 hours on, 12 hours off.
Right.
What else? We had two classes of nutrients, or have what we call methylation adaptogens, and then methyl donor group foods, and we want people to have lavish amounts of both. So we're specifically looking at DNA methylation here, and sweet talking DNA methylation among all of the epigenetic marks.
Right.
So we're very interested in that for a number of reasons.
And before we launch the exact protocol, I'd love to know why you were looking at DNA methylation specifically, and maybe you can tie in a little bit of explanation about DNA methylation, why it's important, and maybe even talk about a little bit about just methylation and epigenetics and globally as well for the people that are coming into this brand new.
Yeah.
That maybe they don't understand epigenetics and what that even is.
Yeah. So epigenetics, epi above, genetics, your genes. Epigenetics is the field of study that concerns itself with all of the myriad biochemical marks that are on and around this, the double strands of DNA, folding it and, marking it to influence what genes are on and what genes are off, what proteins are expressed and what proteins are inhibited. And, this goes back to just how a fertilized egg could develop into a human being of, many, 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 many different cell types. So your eye cell remains an eye cell. It's not a liver cell. It's not a skin cell or heart cell. And that's because of epigenetics. So it's these chemical marks that will turn off certain proteins and allow certain proteins on, to create the unique signature that is you. But there are regions of the epigenome that are malleable and changeable based on environmental inputs. So your retina cell isn't going to turn into a hepatocyte anytime soon. Those are very, reliably inhibited. But, there's plenty of the 30 million marks that respond to how we eat, what we're breathing, our experience in life, our stress, how we're moving our body, and so on and so forth.
Right.
And so this area is where we're very interested. The fact that we can influence potently through our choices gene expression.
Right.
And of all of the myriad epigenetic marks, DNA methylation, and a methyl group, for anybody who doesn't remember it from high school biochem, is a carbon with 3 hydrogens. And when these methyl groups are dotted on a promoter region of a gene, that gene is going to be inhibited from being transcribed. So the protein that gene correlates to won't be produced. If those methyl groups are removed, and we have the machinery to be able to put it down, to remove it, all sorts of stuff, if they're removed, that gene can be turned on. And it's, DNA methylation for a variety of reasons. A is, we can measure it in the laboratory really well and reliably. It's also it's lasting. There's enzymes in the body that will keep the patterns going for a number of different reasons. It's a reliable focus in science than some of the more sort of labile epigenetic processes.
Got it. And so for people listening, basically these are on and off switches for your genes. And then we can look at these on and off switches and what patterns they're in to approximate your biological age using these tests, right?
Yeah, so that's one amazing tool of looking at the epigenome. Yes, so we can identify, like we could measure a fetus in utero and it would be a negative age, or we could look at a centenarian, somebody a plus 100, and see a reliable DNA methylation pattern on the epigenome consistent with that age, and then all points in between. So things change pretty predictably to allow us to see biological age.
Did you see that recent study about, aging accelerates at age 44 and 62 or something?
Yeah.
And was that also using methylation patterns, or was I using something different?
They were looking at multi-omics.
Got it.
Yeah,
that's right. It was something different.
Yes, it was.
Yeah. So and so that highlights the fact that there's a lot of different ways that we can test our biology. And what we're doing now with these tests is that we're using machine learning and algorithms to approximate age based on, I guess, populations and where their aging is for that particular pattern. And so I think the first person to do this with epigenetics was Horvath. Is that right? And so using those algorithms is what you used on your patients to see kind of where they fit into this into biological age.
Yes. Yeah. There is a hotbed of research in clocks of all different stripes.
Now there are, right?
Yeah, that's right. I think that the epigenetic clocks are, for myriad reasons, sticking around and important players on the landscape. But indeed, we're really tussling with this phenomena of measuring aging. It's interesting and it's complex.
Right, but what's incredibly, I think, valuable here is for the first time we have kind of an overall indicator of our current health status in using some of these. And we can use this as kind of a scorecard or a moment in time where we can check our epigenetic age, and then a year from now, check our epigenetic age. And all the things that we did from now till a year from now, how do they add up and stack up in either improving our rate of aging or not improving our rate of aging, right? And so I think that's where we can really use this. That's how I use it clinically, really, with my patients. Is I say, look, this doesn't really mean there's any one specific thing you did right or did wrong, but there are ways that you can utilize this to kind of sum total. How was the last year? I use it every 6 months. How did the last 6 months add up to affecting your biological age?
Yeah, I think it's a great tool. And then I think that we're starting to tease out ones that are, changeable with less time, and we can lean on those. I like the Dunedin pace of aging, which is, such a clock. There's new clocks coming out focusing on different organ systems, and I think those are exciting. It's it's an interesting field. It doesn't replace our standard testing. No. It doesn't replace all of the myriad tools that we have in clinical practice, but it enhances, and it can motivate the patient and really give us insight into how we need to work with that individual.
Right. And what's exciting about now our next topic of discussion here is you've actually figured out a protocol that can positively affect your biological age, and utilizing lifestyle, diet as well, and specific items in the diet. And so let's now go into some of the dietary methylation adaptogens. You said was one category, and what was the second category?
Methyl donors.
Methyl donors. Okay. Yeah. So what's the definition of a methyl adaptogen and a methyl donor?
So they're both. We refer to them as a group as epinutrients, so we know they can influence epigenetic expression. Methylation adaptogens are not, so the donors, actually, I'll start there. The donors help make the carbon and 3 hydrogens. So we've got 30 million of them on the genome. We need these guys a lot. We need to, keep them going, replace them, et cetera, et cetera. And methylation is happening elsewhere in the body. We use it for all sorts of important processes in the body. It's just a carbon and 3 hydrogens. Like I think we were just hardwired to use it because it's ubiquitous, it's easy. So we have this methylation cycle in the body that makes these methyl groups, and we need to keep it humming. And we become less efficient at it diet as we age. So the diet needs to be packed with the nutrients to be able to keep the methylation cycle humming and to be able to keep these methyl groups being produced and available, doing all the myriad important things they do. So the diet is loaded with those foods. And methyl donor nutrients are folate. When you know that folate and B12 are important for you, this is why. Betaine, choline, there's a variety of different minerals that are important, like zinc, magnesium. We know fish oil even has a regulatory role on the methylation cycle. So the diet is just packed with all of those as much as we can. And we could see in our study participants, they raised their circulating methylfolate. So that was, evidence to us that we achieved what we wanted to achieve with the dietary pattern.
Are people on the standard American diet deficient in these methyl groups?
I would say that we definitely see a drop in the ability to methylate as we age. It's just de rigueur.
Got it.
Even when there's a sufficient amount of B12 or folate, but, and you know this as a clinician,
Right.
the reference range for both of those nutrients is incredibly broad.
Right.
We see people with profound B12 deficiency associated, clinical signs and symptoms, but, their B12 is within the reference limit.
Homo reference limit.
Yeah. So same here. But you'll see somebody's homocysteine rise. Really, homocysteine is a standard marker we get in functional medicine routinely, and we always see it rise in people who are, middle-aged and above. And oftentimes we see it in younger individuals as well, actually. It's just not uncommon to see it high. And it's a much more reliable marker of methylation status, B12 status, folate, than measuring them directly. So I would say, we are deficient in these. We don't have enough to maintain, all of the methyl donors that we need. So most of us need more of these nutrients in our lives.
Yeah. So it's important to know, if you're getting blood testing done, if your doctor is doing homocysteine levels, if your homocysteine level is high, it's an indicator that you're probably not getting enough of this in your diet.
Yeah, that's right. An elevated homocysteine. Yeah, that's right.
It's a very important marker. I test it on all of our patients now.
Yeah, it's such a great insight. We think about it as being associated with cardiovascular disease, and it is, but it's so much bigger than that.
So much bigger than that, absolutely.
Yeah.
Okay.
So that's category one.
And can I just, before we move on from that, can you talk a little bit about MTHFR and people that have a genetic deficiency in methylating as well?
Yeah. Yeah. So in those individuals, not all genetic issues like MTHFR variant will show up with problems. Not everybody. And maybe this is because they're getting adequate amount in the diet, or, they're heterozygous, or, they only have one gene variant versus both. For myriad reasons, we don't see everything showing up negatively, but those people, so these are lesions. These are genetic variants you're talking about within the methylation cycle itself. So these are people that could have a problem making that all-important methyl group. I would say preliminary literature, and actually even our own study, we didn't publish on this, but we saw some background evidence to suggest that people with mutations here seem to be potentially aging at an accelerated rate. I want to be nuanced in how I say this because the data are pretty scant. But they need more methyl donor support.
Absolutely.
That's the bottom line.
So these are one of those things that if you have a high homocysteine level, or even if you don't, I think it's valuable to do some sort of genetic testing, because if you can't make these on and off switches at a level that your body needs, it doesn't matter sometimes how much of the nutrient you take in, you probably even need higher levels, or you need methylated versions of some of these nutrients, so you kind of bypass some of these pathways.
That's right.
Yeah.
I would say that's true. It is a continuum. Not everybody is going to need, a really aggressive dose, although some people are. So it is a continuum. But yeah, it's good to know, and it's good to be mindful. I have my MTHFR is fine, but I have, the sister pathway. I've got a variant in that. And my homocysteine, if I'm not really paying attention, will bump right up.
Oh,
So I'm one of those people. Yeah.
Yeah, and I have a few patients like that,
Yeah.
their homocysteine levels are high, and they try to attack it on their own with increasing choline in their diet and taking these more, methyl group rich foods, but they still need to be on methylated B vitamins, I think, sometimes.
Yeah, that's right. That's right. Not everybody, but some of us definitely do.
Right,
Yeah.
right.
Yeah.
Okay. And then the methyl adaptogens, what are
the
those?
methylation adaptogens. Yeah. So there's this cool body of literature showing that phytochemicals, that plant compounds, the colorful aspects of plants, the polyphenols, so things like curcumin, green tea, luteolin, resveratrol, and on and on. There's many. Quercetin. These guys seem to be able to regulate the enzymes involved in DNA methylation. So they're not part of the methylation cycle. They're not making the methyl groups, but they are sort of directing traffic, if you will, like where they're going on the gene to something more favorable. Sure. And we got really excited about this when we were designing the program. And a lot of this science, so this was against cells. We've got some early human data now looking at it from our group, but it was cancer. So we could see, cancer would shut down genes that protect us from cancer, from getting cancer. They shut them down by putting these methyl groups on. And these beautiful polyphenol compounds, many different varieties of them, will turn those genes back on, allow them to kind of get back into, protecting us from cancer. Yeah, just right there, changing methylation on the DNA.
That's amazing.
And so every forkful of food in the dietary pattern is both of these. So every single forkful is packed with these epinutrients directing traffic.
I love that.
And we believe, and I think the evidence now is coming out that this, that it was a wise choice for us to focus here because, more and more scientists are suggesting that, it's really, if you look at all of the hallmarks of aging, the epigenome is sort of the business. It's like the first step, and if you can get in there and change the epigenome, you can change all of the hallmarks of aging. And I know everything's very interconnected, but, it makes me excited and heartened about this dietary pattern that we just happened to start, with epigenetics.
Absolutely. I think David Sinclair truly believes that too, that the epigenome is really kind of the centerpiece of all the hallmarks of aging.
That's right.
And so that all leads back into epigenetic changes.
Yes.
And going back to a forkful of methyl adaptogens and a forkful of methyl donors, talk a little bit more about the actual diet and how do we get a forkful of these at home?
Yeah. Like a well-designed salad, it could be a forkful of both. Yeah. So again, it's vegetable intensive. Right. There's somebody who Julie Gibson. So she's somebody who's sort of been a rock star in biological age, reversing her own biological age. And she uses our diet. And so she perceives herself as needing to eat a pound of veggies a day. And she talks about how she does that and smoothies, et cetera. But, lots of greens, lots of cruciferous, and lots of colorful veg. Right. So if you can just get that in, all of it is dense with information. That right there is full of methylation adaptogen and methyl donor nutrients. And then layer onto that mushrooms, like any kind of mushrooms. I love shiitake, but maitake, turkey tail, even button, the lowly button mushroom has a place here. Yeah, all of them. They're, and it's a superstar because it's both this, it has the adaptogenic potential and it also has the methyl donor part of it as well. Good quality protein is essential. Good, clean, fatty fish, chicken, whatever your protein of choice is, getting a decent amount is essential for the methylation cycle. And, it's important for myriad other reasons as well. Methylation adaptogens are also the spices that we routinely use. Curcumin, thyme, rosemary, on and on. We've got 30 pages of methyl donor foods and methylation adaptogen foods. It's my favorite part of the book, this epinutrient appendix. Right. And you can go in there and just see just almost everything, almost every quality food, whole food, has some element of epinutrients. And so we just really try to pack the diet full of the ones that have the most bang for your buck.
Yeah, yeah.
Green tea,
And for people that want to get this dietary program, they just buy the Younger You book, which is the one that I used, and has the entire diet kind of laid out in the 8-week program.
Yes.
There are some supplements, though, that can be used as well in this methylation, adaptogen methylation donor program. What are some of those?
We used a probiotic. Lactobacillus plantarum. It's got some data behind it as being able to support the production of folate from the microbiome. So our microbiome, a healthy gut microbiome, makes a multivitamin, basically. I know. It's pretty extraordinary.
Pretty amazing.
It is. And the diet is packed with prebiotic food, so it's going to really be stoking a healthy microbiome. And then we layered into this a probiotic that we were hoping would help increase folate. And we did, in fact, increase circulating folate. But the diet is folate dense, so we can't say it was one or the other. We also included a greens powder. I'm incredibly bullish on the methylation adaptogen category of epinutrients. So we just packed a little bit more in with a greens powder.
Got it.
Yeah.
And I remember also there was another supplement that was it like a liver supplement or an organ meat supplement?
Yeah. So liver, we're huge fans of liver. I mean, we really are. No one wants to eat it, including myself. I don't.
But it's easier to swallow in a pill.
It's easier to swallow. It's a multivitamin mineral in a food matrix.
Right.
We really believe in this food matrix. So as we were talking about earlier, of course, these, vitamins are essential. I take plenty of vitamins myself. But as much as we can possibly get in a food matrix, it's designed for bioavailability. There's accessory nutrients we don't even understand. There's interactions. There's so much to the complexity of food that we have to respect and lean on it. So liver. It's the element of our diet that was least adhered to in both of our publications. But we now use supplements. Like we just in our first study, I wasn't aware that you could get these, freeze-dried liver capsules, and they were super clean. And I would have had our participants use those. We instead tried to get them to eat liver, and then on adherence data, it was terrible. Nobody really wanted to. But the capsules are super easy, and I love them. I think it's an awesome way to get your multivitamin.
Yeah, it's like you said, that food matrix is incredibly powerful, and we don't even know how powerful. And so when you take vitamins as, nature gives it to us in either like a liver capsule or eating vegetables themselves, there's such a synergistic effect there. And what we've done with the standard multivitamin is we've broken everything down into individual components and put it together again. Yes. And we're missing a lot of stuff.
We're missing a lot.
Right.
We are missing a lot. Yeah, we might think, and vitamins have their place. Multivitamins have their place. You'll get no argument. But the level of complexity that, a well-designed salad has on that, it's
mind-blowing.
Beyond.
Beyond. Okay, so that's the food component / supplements. And then let's talk about the lifestyle components of the Younger You program. Can you talk about those?
Sure. Yeah. So we wanted people to be exercising. Exercise is an epinutrient as much as any of the foods. It's an extraordinarily potent regulator of optimal gene expression, as you know. It's just so powerful, and the data are so strong. It can act. So going back to our earliest entry into thinking about epigenetics, it was cancer and how the tumor microenvironment turns genes off for its own growth, turns genes on for its own growth. So it just really manipulates. And we can actually look at all of the chronic diseases of aging and see similar patterns. There just happens to be the most research in cancer. Well, exercise acts like those epinutrients that re-express tumor suppressor genes. Like it just gets in there and changes gene expression dynamics towards something more favorable. It's beautiful and it's so powerful.
Right.
And, the one study that I think I love the most looking at this is one that demonstrates it's more potent the older we get. So we'll get more epigenetic change more
that's good to
epinutrient
hear.
bang for our buck. I know, I know. Yeah, so exercise was an essential part.
Was there a specific type of exercise?
No, so we wanted this whole program to be broadly adaptable.
Okay.
And so we want people to be able to, manipulate and do what they want as much as possible. So we just want people to do a minimum of 30 minutes, 5 days a week. So it's a program for people who aren't exercisers. One who doesn't have a decent exercise habit can step in and do this.
Yeah.
We want people to achieve an exertion of 60 to 80% of their maximum. So 60% you can still carry on a conversation. 80% you could still have a conversation, but you're breathing heavier. You're probably not talking quite as much. So do whatever you want. Achieve that 60 to 80% of perceived max exertion, for a half an hour.
So that's like zone 2.
Yeah,
Or a little bit more than
it's
zone
a little
2.
bit more than
Okay. Got it.
So that's the foundational intervention for our study. That's what we did in our 2 research publications. But clearly you can turn the volume up from there. So a younger you 2.0 could include resistance training, high intensity interval training, a combination of those that we know have really strong data on them.
I love it.
Yeah.
So you had the exercise component, the dietary component. Were there any other components?
Yeah. So we wanted people to be sleeping well. So
Right.
sleep and stress reduction. Sleep, we had nutrition coaches working with our participants, meeting with them weekly, and they would just dialogue about sleep hygiene and, good sleep tips for, making sure you have enough time. I mean, I would say the biggest limitation to me getting adequate sleep is really not going to bed on time. I don't give myself enough runway to achieve 7 hours. 7 hours is like my sweet spot. And just, is the room dark? Is it cool enough? Is there noise, et cetera? The usual. We recommend magnesium in the book, a little bit of melatonin, maybe some glycine as needed for sleep. But if you're not sleeping, you're definitely accelerating biological age. It's really that simple.
Absolutely.
And those data are in humans.
Right.
And if you're sleeping well, you're preventing all of those things. So, sleep is another exquisitely important epinutrient.
Sure. On the stress reduction front, were there any specific techniques?
Yeah. So if you're stressed out, if you're in that toxic stress, you're aging, unfortunately.
Yeah, absolutely.
And we can see that via the chronic diseases of aging, but we can also see it's really anchored in these negative epigenetic changes. Stress is quite toxic on the epigenome. So we wanted people to do a simple relaxation response, a basic breathing exercise, 10 minutes twice a day, ideally, created by Herbert Benson many years ago out of Harvard.
Okay. And can people find this on the internet?
Yeah, they can find it in the internet, and it's in the book as well. It's just it's super basic. It's interesting. I think he created it in the 70s, and it was at a time when there was sort of pushback on meditation. So he called it the relaxation response. But that's basically what it is. It's meditation. Now, that's what we used in our study. But there's many ways to get into the zone.
Right,
One could do yoga.
Yeah.
One could do Tai Chi. Like there's just many, many ways that we could do that. So we've studied the relaxation response as created by Herbert Benson, but really any kind of exercise to get grounded, yoga nidra could be really important and helpful.
Got it. So the people in the study underwent these dietary, relaxation, sleep, coaching, I would say, and 30 minutes of exercise 5 times a day. That was one arm. And then it was a randomized study, right? So the other arm didn't do these things?
Correct.
They just had a normal lifestyle.
They lived, that's right.
Their normal lifestyle. Okay. And there was a biological age reduction measured by methylation clocks of how many years in your patients?
Compared to our control group, it was over 3 years.
Yeah. And 8 weeks.
In 8 weeks' time.
So
pretty exciting.
Weeks turned back biological age by 3 by 3 years. It's incredibly powerful.
Yeah.
And you didn't use any hormone manipulating drugs or any of these other things. And so I think that just goes to highlight how incredibly important this pathway is and how, to David Sinclair's point, it could be the kind of the center point of all the other hallmarks of aging.
I mean, so we built the dietary pattern is good for many reasons,
Right.
but it was really built brick by brick to influence epigenetics. And and the other elements were brought in because of their known influence on epigenetics. So focusing on that, focusing specifically on DNA methylation seemed
like the right
pretty
move.
cool.
Yeah, you wrote the first book on this and why it's important. And I think many people have used your protocol since then and have seen their epigenome reverse. And now you can get these tests pretty easily.
Yes.
Just from a finger stick blood test you mail in now, right?
Yes.
So people can do this test on their own before and after the program.
Yeah, it's fun. Yeah, and we have groups going at our clinic. Quarterly we'll have little groups going, and you guys are going to be doing groups. It's fun. It's a nice program to do together. Especially if this is a change from your usual dietary pattern. It's nice to do that. Have that really juicy community support, which is an epinutrient in and of itself as well. So it's nice to add that element.
Yeah.
But it was really quite exciting. And then the within group comparison. So the guys who underwent the intervention at baseline, their epigenetic, we measured their epigenetic age, and compared to themselves at baseline to follow up, their bioage reversed by just over 2 years. So the within group comparison was statistically significant at just over 2 years, and then compared to the control, we saw a little bit stronger response. But very cool.
Incredibly cool. And I think you mentioned the community aspect of this. I think that's incredibly important as well.
Yeah.
Like you said, it's an epigenetic
it is.
modifier as well. And the groups that you run are through telemedicine. So anyone can do this from anywhere pretty much, right?
That's right.
Yeah. And so all they need is a book, a computer, and off you go, reverse your biological age by 3 years.
That's right.
Which is fantastic. We're super excited to do this as well in our clinic.
Yeah, it's awesome. And I should just add, I mentioned this in the beginning, but in our research study, we saw, not only did we see circulating folate increase without supplements, triglycerides dropped, LDL dropped, weight, people lost weight, a significant amount of weight, and we didn't report that in our first study. We have a paper in peer review right now where we talk about the weight change. In our clinic practice, we see inflammation drop. I mean, we just really see lives changed using this intervention.
Yeah, I mean, dropping inflammation, dropping triglyceride levels, all these immediate blood biomarkers of actual, not just age reversal, but chronic disease avoidance is incredibly important.
Yeah.
So I think this is really important it's important for everybody, but really for people that are looking to make a change relatively quickly in their biology. Yeah. And maybe, do you almost look at it as something that someone who needs a jump start can do something like this?
Yeah, for sure.
Yeah.
Yes. In our practice, we used it with some people who were really, struggling. In fact, originally we were using it with that very sensitive population, and I'm sure who can't tolerate anything. You give them your most gentle vitamin and they have a reaction to it. And when we originally unrolled this, we were using it with that group and people responded.
Wow.
I know. It was amazing. And they responded profoundly. And if they really hung in there with it, we would see remarkable change, not only in how they felt, but in their various biomarkers. It's an awesome jumpstart. It's an awesome way to get engaged in this. And then after you finish the 8 weeks, you can take elements and incorporate into your regular lifestyle. You can continue to use this epinutrient information. Like, you will never eat a forkful of food without thinking about what kind of information you're putting into your body. And do you want to actually give your body that information or not? It's just, there's such a different and empowering relationship you can have with food and with your lifestyle choices.
Absolutely. I mean, I think, as far as the testing goes, let's talk about that real quick. You don't need to do the testing to put this into play in your life. You'll actually feel the difference after 8 weeks.
Yeah, that's right. So in the book, 2 cool pieces. We have a bioage quiz that people can take that's in the book, or you can just go to my website and do it for free there. You don't even have to get the book. And we have found, cool enough, and this will hopefully be another paper or white paper or something, that it correlates significantly with the Dunedin pace of aging.
I mean, that's amazing.
I know. We've got a group of just over 70 individuals who've done both our quiz, plus the pace of aging, and we can see that it tightly correlates. So the quiz actually has a little bit of evidence behind it now that, that it's not a bad quiz, that it's not just a pop quiz.
And this is a quick, 10-minute quiz from what I remember on your website.
It's
and
easy.
you get the score right away.
You get the score right away.
Scores. So this is a great way to test yourself, and costs you nothing except 10 minutes.
That's right. Yeah. And then there's something that you use in practice as well called the Multi-Symptom questionnaire, the MSQ. Sure. Mm-hmm. And we have that in the book as well. It's the smartest thing. If you're going to do this whole program, do an MSQ at baseline. Right. It's just a careful exploration of your various symptoms, and then do it at the end and see what the difference is. Mm. Chances are really good that your score at the end is going to be lower.
Sure.
It's just that, it's that simple. People feel good on this dietary pattern.
Yeah, that's
so
fantastic.
Those aches and pains, the headache, the allergies, we can just see the GI stuff. We can see that clear.
Right, which just goes to speak to the functional kind of root cause methodology of how this program is affecting our health. Because when you affect the root causes, inflammation, epigenetic expression, et cetera, can lead to less symptoms all over your body, and whether it be migraines or, like you said, joint pain, et cetera, this is really working at a root cause level.
Yeah.
Right.
Yeah.
Amazing.
We think so.
You had mentioned something a little bit earlier where you're going to do Younger You 2.0, or you've done Younger You 2.0.
We have iterations of Younger You 2.0. So, having the advantage of using it in clinical practice for a long time, we know how to do a Younger You. We know how to individualize it for the person sitting in front of us, to make sure that it's just the best dietary pattern for them. So maybe they need to turn the ketone volume up, or maybe they're working on muscle mass, and we can tweak it. Or maybe they've got inflammatory bowel disease and can't tolerate certain foods. So we've really learned how to individualize it in clinical practice. But we're excited also about turning the volume up ever so slightly on protein, and really supporting people in doing resistance training along with the Younger You. So that's a 2.0, maybe turning the volume up a little bit on that intermittent fasting window, because we know the research on that is just so strong. The other piece that we learned recently, a group of scientists in the UK out of a group called NetMind undertook the program themselves in the middle of the winter in London, so dark and rainy, and they did a full supplement washout. They stopped all of their supplements 2 weeks before and continued off of them through the duration of the study, and all of them became vitamin D deficient. And we don't want that. In our original study, and unfortunately they missed this, people who came into it on vitamin D were allowed to remain on their vitamin D, but the research on vitamin D as deficiency as an age accelerant, and then repletion as an age decelerant is, study after study. It's pretty strong at this point. So even though this group got younger as measured by the pace of aging, that's awesome, they all became D deficient. So who knows? If they didn't, maybe they'd be even younger. But my lesson is D will be a part of the pattern going forward.
So that'll be a supplement that should be continued throughout the entire thing.
That's right.
Got it. Are there any other supplements that you would continue through the program, like an omega-3 supplement, for example?
I think that's fair. So in our first study, we had them do a washout, but in the second case series that we published on women, we just had them stay with their supplements. So whatever they started on, they continued on. And then I cover a lot of those foundational supplements. And of course, magnesium, fish oil, are key players. So yeah, I mean, we'll see. You will see. We're working on funding right now to do a few different arms of Younger You iterations, but yeah, those are really important nutrients.
Yeah, and also I think it'll be interesting to kind of suss out where most of the effect is coming from by doing all these different arms.
That is a paper that is in peer review right now.
Oh, okay.
So we did do that actually. Somebody, it's pretty interesting. And we have an idea. I mean, the reality is the overall dietary pattern and the lifestyle changes, they're all good and they're all really strongly anchored in good science. But it does seem like some of those so-called methylation adaptogens, those polyphenols, were extra important.
Wow.
I know.
That's so
it's pretty cool.
incredible to know because, I think as far as polyphenols go in the standard diet that I see most people eating, I mean, there's very few polyphenols in people's diets.
Yeah.
And I mean, you have certain people doing things like the carnivore diet that are getting 0 polyphenols.
I know.
And so there might be, I wonder if anyone's ever done any epigenetic clocks on people on the carnivore diet.
That's a really good question. Yeah.
Right.
And like if you're a diabetic and you start the carnivore diet, if you're diabetic, your age is accelerating. I mean, unfortunately, any chronic disease, we're going to see age acceleration. But diabetes is really, I think it's on average, Nathan Price published on it being like 6 years old.
Sure.
Like it's definitely an age accelerant. If you go on the carnivore diet, you're going to drop your sugar, you're going to drop your insulin. So it's a short-term intervention.
Agreed.
So in that setting, you would probably see a biological age deceleration. You would see a beneficial change. But measure that same person, a year from now when their microbiome has radically changed. And, my guess is that we might be seeing, it creeping forward. And we're going to see micronutrient deficiencies and all sorts of stuff.
Absolutely. I mean, I'm not knocking the carnivore diet at all. I've seen a lot of success in a lot of patients with it, not just for diabetes reversal, but also for weight loss, et cetera. Yeah. But I do think, it's so individual. You have to do an N of 1 experiment on yourself when you're going through these experiments, and having tests available like blood biomarkers like methylation, DNA tests, et cetera.
Yes,
I think people need to look at those on an individual basis and see how these different interventions are affecting them.
That's right. That's right. And then the long term, the long term outcome of a carnivore dietary pattern, I just, I think it's unsustainable. And I think, to your point, you're going to really aggressively change the microbiome. And so the microbiome is acting on those polyphenols and producing all these postbiotic compounds that are just wonderkins. Yeah. They're just doing all sorts of extraordinary, important work in the body. Yeah. And you won't get those, and you'll get a die-off on the bacteria that are actually producing them, and the whole ratio will change. So I think for the long term, we're looking at a difficulty with that. But I'm a believer in the N of 1.
Absolutely. Are you testing the microbiome before and after the Younger You program?
In the clinic, that's a standard part of it. And you do see in somebody on a carnivore diet a more pro-inflammatory pattern. Especially, I should say, carnivore diet, over a period of time. Yeah. And you see, favorable changes on a veggie dense diet. Interestingly, there was a study, you saw the omnivore and the vegan study that got so much PR.
Right.
And they saw favorable short-term changes in the vegan diet. They lost a little bit of weight. Their clocks seemed to slow down relative to the omnivore. But same kind of complaint with that dietary pattern. Like for the long haul, you're going to see micronutrient deficiencies. You'll see a change to the microbiome and so forth. Yeah. So in the short term, some good benefit, but some possible fallout with long-term use.
Yeah. We this dietary debate will probably go on past our lifetimes.
Yeah. That's right.
But, I think once again, it's N of 1. Like somewhere in the middle is probably right for most people, where you have that Mediterranean diet with, a good amount of vegetables, good amount of protein. Yeah. All has to be well-sourced, of course, and some good fats. And then some people are going to do better with more vegetables. Some are going to do better with more protein. That's right. And maybe there's different points in your life where you do better on one and the other one too, right?
That's a very good point. Yeah, I mean, if you're doing a bunch of high-intensity and interval training, you're going to need some readily accessible carbs. Right. If you kick those to the curb and you're just doing, something with almost no carbs, like carnivore, you're going to feel that when you really try to hit those heights with exercise. So to your point, yes.
Your days you're doing your two-hour bike rides, I'm sure you need some carbs.
Well, it just made me think of when I was competing. There was a burrito shop around the corner, and I'd always have a big burrito with the wrapper and chips. Right. That was my night before my race meal.
The night
but
before
those
the
chips
race
were
meal.
really essential. I needed that. Yes.
I love it. Yeah, those night before the race meals are always
they're good.
They're really good. Kind of like just eat as much as you can.
Yes.
That's awesome. So you're still in clinical practice, right? Are you still seeing patients as well?
I am. I have a micro practice. I'm keeping my toe in the pond. But I do.
I think it's important. I do the same thing.
Yeah.
It's always good to see patients and see how different people react differently.
Yes.
I learned so much from patients. And that's why they call it a practice.
I know. It really is. It's important to kind of just to keep our head in that game,
right?
As providers. So
exactly.
Yeah, I do.
And you're one of these people, Kara, that you're always keeping your finger on the pulse of what's going on in the research. You're so well read. You're looking at the research all the time. What are you seeing as some of the things that are really piquing your interest in biological age reversal?
Well, I mean, I know that you're big on therapeutic plasma exchange, and I just had my first one. I'm so excited. I think the research coming out on it is ridiculously exciting, and I think it's a safe intervention. There's just so many pluses to it. I have clinical experience seeing some extraordinary turnaround in some of my patients with inflammatory conditions, but doing it as a wellness intervention, I'm pretty excited about. But aside from that, so there's a few things that I'm very interested in. One is there's a clock that isn't available in the clinical setting. It's in the research setting that appears to capture all of the hallmarks of aging.
Oh.
So a lot of the clocks that we're using now, if not all of them, are surrogate markers. So they may not be capturing the aging phenomena itself, but they're suggesting it. But this clock, which is out of Vittorio Sebastiano's lab,
Sure.
of Vittorio.
He's in San Francisco, right?
Yeah. Mm-hmm. And this really seems to be capturing. So when there's a favorable influence on the clock, it seems to track with all of the hallmarks. And the intervention that does it in their laboratory is a Yamanaka factor intervention. So it's Yamanaka factors, as you know, directly addresses the epigenome. So it's another piece of evidence, and it's similar to what David Sinclair is doing in his lab. So they're changing gene expression vis-a-vis the epigenome using these Yamanaka factors. And heavily DNA methylation is there. They're changing DNA methylation, and they're able to see all of the downstream hallmarks change. They also demonstrated that caloric restriction, this is still in animals, so we need to get it into humans, had similar changes to the Yamanaka factors on this particular clock, which I think is really exciting. Yeah. And looking at the elements of the clock, there's some evidence that these beautiful polyphenols might play a role and might be able to favorably influence the clock. So I'm excited to hopefully, have access to that clock in our research. We totaled up the phytochemicals, the polyphenols specifically in our intervention. There's about 3 grams, which is really, it's kind of dwarfs any other sort of polyphenol-dense dietary pattern.
Sure.
And I would like to do some work with that clock to see, like, are we touching, is this dietary pattern at all touching on, what the Yamanaka factors are doing?
Yeah.
I know that Vittorio Sebastiano is sort of jumping the FDA hoops to bring a Yamanaka-based intervention to clinical trials, which is ridiculously exciting.
That's exciting.
Mm-hmm. And it's going to start with skin. I'm like, I'll do it. I'll volunteer. So we need to, as Sergey Young talks about, he has a longevity fund that invests in all sorts of, including the X Prize, all sorts of really cool businesses in the longevity space and biotechs and so forth. He talks about the longevity bridge. We need to get to the other side when some of these interventions will be ready, and we need to be taking care of ourselves now. We need, therapeutic plasma exchange using some version of the younger you dietary pattern, exercise, adequate sleep. So all of those things I think are important. I'm bullish on rapamycin, quite frankly.
Yeah.
I'm less excited about metformin, I think, for some of the negative fallout. But I think there's interventions we have right now that will take us a long way, over the longevity bridge until we get to really some of these exciting interventions that scientists are working on right now. So it's a good time to be alive.
I agree. I think the Yamanaka factors, for the listeners who don't know what they are, they're a Nobel Prize winning discovery of certain proteins that can actually turn around or reverse cellular aging. We have no control over this process other than to give 1, 2, or 3 of the Yamanaka factors. And so there's a lot of research that needs to get done.
Yeah. Actually, they time it. So there's, and they can shut down. So that's kind of the delivery of the Yamanaka factors and, the intervention that will time it and stop it. Because it will, if you, left to its own devices, it will take a somatic cell. So it will take that retinal cell or, a cardiomyocyte and restore and turn it back to pluripotent stem cell. So it's like as young as you get.
Right, exactly.
And you don't want that.
You don't want that.
So there's a lot of energy going on the delivery and the intervention used to stop it and how long the exposure is and all of that.
But it's almost like science fiction type of discovery. I mean, you think about that, like I could take a skin cell from you and turn it back into a pluripotential stem cell. And, David Sinclair's talking about then taking that and cloning that into another you. I mean, there's like, we're in the realm of science fiction right now. And obviously some of it's like a little bit overblown, but just the fact that we're there and people, this is legitimate science. People are winning Nobel Prizes for these things. I mean, there's work to be done, which is incredibly more exciting than we just don't know. And there's no work to be done because we have no direction to head in.
It is. It's very exciting. It's super disruptive.
Right.
I think people are taking to heart the fact that we spend, our final 16 plus years sick. With multiple diseases on polypharmacy. I mean, it's dismal. Right. The health trajectory or the disease trajectory that we're on is depressing. But the sun is right there. I mean, the possibility is right there, and people are getting it and taking charge. And to your point, when we were talking off air earlier, the mission. I was actually reading the mission statement of Next Health over there. It's just really cool. You want to disrupt this whole, like all of us want to disrupt the slumber that the medical system is in this country and really the world. So, it's exciting. And the possibilities they're science fiction, but, just very exciting science fiction.
Yeah. The key is to stay healthy long enough and alive long enough to see what science fiction actually comes true, I mean, we stayed alive long enough to see a self-driving car actually work. Haven't you taken a Waymo yet?
No, I haven't.
You can get them in LA right down the street over here. Well, it's a car that'll drive you around Los Angeles with no driver.
How do you like it?
It is actually really cool. I mean, it's science fiction come to life, and these things are happening during our lifetime.
I think, when Vittorio Sebastiano was talking about it and it was just the way that he structured it. So, a lot of us think we're going to take a cocktail of Yamanaka factors and, turn back to 20. And that is, out there and likely won't land. But rejuvenating the ovaries, exposing the ovaries, because each organ system is different. It's going to need its own cocktail and its own time frame, et cetera. Rejuvenating the ovaries in a woman, allowing her to, make estrogen for longer and progesterone and so forth, will take care of, not just ovarian function and, her fertility, should she want to have a kid, but, bone health, skin health, brain health, heart health. So it's like this local application in an organ where you can get this full system leverage effect, I think makes sense to me.
Absolutely. And there are scientists that are injecting Yamanaka factors into livers of animals and causing liver regeneration, which could be incredibly helpful for people that have maybe fatty or fibrous liver disease, right? So these kind of things can be therapeutic in the near future.
Totally.
Yeah, it's so exciting.
It's very exciting.
Right. And plasma exchange is exciting because it actually, for those of you who don't know what that is, that's a therapy that can be done to remove your blood, 150 cc's at a time, remove the plasma and then re-inject it into the other arm, your blood cells. But in the plasmas where all the negative toxins and the cytokines, that inflammatory cytokines live, and just kind of removing it and giving your body a break from these toxins and cytokines can allow your body time to heal. And so these are therapies that have been FDA approved, that have been in the hospital for a long time, that we're bringing out into the wellness and health care world, the health world. And I think using things like the Younger You program combined with plasma exchange all together can keep us healthier long enough to experience some of these incredible mind-blowing technologies. But, these things cost money, but what moves the needle more than even all of the things that we talk about that cost money are the things that don't cost money and putting those into your lifestyle first.
Yeah, that's right.
Right. Yeah.
Get a good night's sleep. Do a little meditation. Get your body moving. Give someone a hug. Yeah.
Right.
Yeah.
What I love about the Younger You program is it's also free pretty much. I mean, you can do it for free. You can get the book. You can take your test online and institute it into your life.
Yep.
And it is a sustainable program, actually.
I think it is.
Right.
Yeah, you can transition the habit and just transition into that and sort of get used to the targets, and it is sustainable and it's nutritionally replete.
Yeah. It's fantastic. Well, thank you for all the work that you're doing, Dr. Fitzgerald, and more to come, I'm sure.
Yes.
The next time you're in Los Angeles, I'd love to have you back on the show to talk about the new things that you're doing.
I'd love to be here and go back to the clinic. It was so fun.
Thank you, Dr. Fitzgerald.
Yeah, absolutely.
Oh, one more thing. Where can people find you? Tell us all the ways.
You can find me. You can find my whole team at drfitzgerald.com. Actually, no, let me say it again. It's not my website. You can find me. You can find our whole team at drkarafitzgerald.com.
Right.
Super easy.
And doesn't youngeryou.com work as well?
Yeah, youngeryou.com. Yeah, you can get to
That
the
takes
book.
you to the book
yeah.
and the testing. And drfitzgerald.com is where people can interact with your clinical team.
Yes.
And if they want to join a Younger You intensive program through telemedicine, how they can do that also on drkarafitzgerald.com?
Yes, and they can access the Younger You world through drkarafitzgerald.com. So it's if you need to remember just one, just drkarafitzgerald.com.
Right.
Newsletters over there, recipes, et cetera, et cetera.
Yeah, your newsletter is great, actually. I really
Thank you.
enjoy it. Thank you so much for joining us today.
Thank you. It's great to be here.
Thank you.